Hi, this is E. David Crawford. Joining me at Grand Rounds in Urology on a topic called radioligand therapy is Dr. Scott T. Tagawa. Scott is the Richard A. Strenton Associate Professor in Hematology and Oncology and an Associate Professor in Clinical Medicine and Urology at the Weill Cornell School of Medicine, Columbia in New York. He got his BS degree from Georgetown and then went to the West Coast at USC, did his medical school there, internship and residency, and has since returned to New York. I'm excited to have Scott with us today. He is clearly an international leader in this whole area of radioligand therapy, theranostics, or whatever you want to call it. And he's going to share with us in the next couple minutes an overview of this and how it integrates into prostate cancer. Scott?
Thank you very much for that introduction and the invitation, David. I'm very happy to talk about what I consider a very hot topic in prostate cancer. I have some slides that I'm going to be showing. Some of them I'm going to skip over quite quickly because they were designed for a recent PSMA summit that was held with a number of United States institutions from a couple in New York, Baltimore, and Los Angeles, as well as folks from Bayer and Germany and Australia, where we got together to talk about where the field should go forward. But it is a quite hot topic in prostate cancer right now. So there's some general disclosures that I have, and another disclosure is that some of the work here is being funded by the Prostate Cancer Foundation. So two points of interest in terms of background. One is the target, and one is the inherent general radiosensitivity of the disease of prostate cancer. So PSMA, which stands for prostate-specific membrane antigen, is more or less present on all prostate cancer cells. Now, it's not exactly true, particularly in later stages of disease that might lose the androgen receptor pathway. But as we know, and the reason that we have used the term castration-resistant prostate cancer is that the androgen receptor pathway remains intact throughout a patient's life cycle with the disease. And basically what this slide shows is that as long as the androgen receptor pathway is intact, PSMA is there. That's point number one. And point number two, as AR signaling is present but dysregulated, as happens in castration-resistant disease or with treatments with drugs such as abiraterone and enzalutamide, PSMA expression on a per-cell basis goes up. So as we've gone into this new era with new more potent hormonal drugs, PSMA remains a target of interest. I think most people generally know that prostate cancer is a radiosensitive disease. We cure patients every day that walk in the door with prostate cancer that's only within the prostate. We can also salvage patients after surgery and palliate those with metastatic disease. We also know that a dose-response relationship exists, so higher doses lead to better outcomes, including higher cure rate. And we also know, and the third point is be important as I talk about our current strategy going forward, is that repopulation is a mechanism of resistance. So, and that's not that different with radiation or chemo or other drugs. The point is, you give one dose, or with antibiotics, for instance, you give one dose today, wait a while, give another low sublethal dose, and that's a way to promote resistance. That's a way we create resistant cell lines, and that's what happens in patients. That's really the background of our approach. Over more than a decade, we've done a number of sequential trials using a PSMA antibody, and I'll get into the difference between antibodies and small molecules. They're more or less summarize each individual study here, but really what we found is that the higher the dose that we're able to administer, the better the outcomes are, but also with increased toxicity, as summarized on this maybe somewhat complicated slide. But basically, the higher the dose, the more PSA goes down, and the longer patients live. So it's both PSA declines as well as overall response, which is another, and this is a summary of what we've done over the last decade or so, and that's really why we designed the current clinical trials in the way we have to really optimize the dose that is able to be delivered, number one, and then we also look to optimize the patient population. So as many people know, PSMA has been a target that has been of interest for a couple of decades, and has been in the clinic for a couple of decades, initially with imaging and then with some of the treatment studies that I showed. There are a number of small molecule or ligands that are available. They're mostly urea inhibitors. I don't think we necessarily need to go into those details, but they've been around for imaging as really standard imaging agents in Europe and other places, available investigationally and a little bit more widely in the United States. But we've also been able to radiolabel these agents and or attach drugs. The current laws in places other than the United States are when patients have refractory disease, nuclear medicine physicians can more or less do what they want to do as part of standard of care, but they can't necessarily do it as part of treatment. So there's a number of publications, more than 25 publications, that look to be prospective clinical trials at a glance, but they're not. But the first prospective clinical trial data was presented by our Australian colleagues at ESMO, and this is a summary of the data where they gave a modest dose and then were able to deliver subsequent doses to a selected patient population. So they, I'm not going to go into details, but did PSMA PETs, FTG PETs, and decided which patients were going to be more optimal for treatment, and this is their initial patient population that's treated. They've now finished their overall trial, and I expect to hear results sometime in 2018. Like I said, what was exciting for me is that this is, or was, the first prospective study ever done. So with all that background, what we've taken is our background with the antibody, the existing data from mostly Europe, but Australia also, in those individual patients, which likely now are 1,000 or more. And we didn't want to start from scratch, but we wanted to assess things in a prospective way. So we've started a series of 3 new clinical trials that are starting initially in New York and are soon to open at a couple of the places. And I'll go through quickly these individual trials. So no one has ever done a dose escalation study of any of these small molecule compounds that have been radiolabeled with anything, whether it's an alpha or a beta emitter. So the most common, in my viewpoint, small molecule that's been used is something that's called PSMA-617 that came out of Heidelberg. The most common radionuclide that's been used is lutetium-177, which is a short-range beta emitter. For those of you that are not sure of the difference between beta and gamma, it's a relatively weak compared to alpha emitter, but can travel a longer distance. Lutetium is similar to iodine, is that in modeling studies is optimal for tumors that are a couple of millimeters rather than centimeters, like a longer emitter such as yttrium. That being said, this is a relatively standard patient population for these types of drugs, and that includes, this is basically the population that radium was developed, although we didn't have as potent air signaling inhibitors at that time. So, the patient population's here, and basically what we're doing is we're taking our prior decade of experience plus the current German experience and saying, well, dose fractionation allows us to give a higher dose as well as a higher dose intensity. So rather than going back to that repopulation resistance data, we can give a much higher dose that should be in tumors for a minimum of a month, if not longer. So to retain a higher dose to decrease the chance of resistance. And then we're watching them. In prospective, we're watching them very closely. So weekly times 6 every other week, and then monthly, to get a real sense of what is going on in both an efficacy and especially a toxicity point of view. And then we're repeating, essentially per prostate cancer working 3, standard imaging. We do happen to have pre and post treatment PSMA images, and this is, an ongoing study, and a number of secondary endpoints that are there. I'll show you an anecdotal response, but this is where we are today, and we're finishing enrollment in cohort 4 which is about 2.5 times a dose per cycle than is typically given in countries like Germany and Australia. This is just an anecdotal experience. I'm not ready for prime time, but it is comforting in that we haven't seen dose-limiting toxicity, and like other places, we've seen efficacy, which is not that surprising. We have been using for a while monoclonal antibodies, and basically the major thing that is different is, frankly, their size. And because of their size, they have different properties in the body. Antibodies are circulating for a lot longer, so that may be better because they're delivering radionuclide or drug or whatever's attached to it on day 2, day 3, day 4, day 5, etc. But that is at the expense of continual circulation through the marrow, and that is why we see dose-limiting toxicity of myelosuppression anytime we use radioimmunotherapy. That is, a radionuclide attached to an antibody. The small molecule ligands are about 100 times smaller, and the advantage/disadvantage is the short circulation time. So a few hours is how long they're in circulation. They rapidly go to sites of PSMA expression in this case and are excreted in terms of the kidney. So the advantage is they're not flooding through the marrow that long. However, they only have those few hours to get to the tumor, and then that's it. And then because of their size, they are able to have access to sites of PSMA expression that antibodies can't. And clinically, what is the problem so far is salivary glands with xerostomia or dry mouth that leads to problems with taste and teeth falling out eventually if it's a potent dose. And then in theory, at least, and what's been seen in at least one preclinical model is kidney toxicity. So PSMA is expressed to a small degree on the luminal aspect of the proximal renal tubules. What does that mean for a patient or clinician? There may be radiation delivered to the kidney with these small molecules. Luckily, we have not seen clinical renal failure so far, but it may be a delayed effect. Because of that, these different properties and this preclinical data that we have here that shows that there's different binding sites and that the amount of radionuclide or drug or whatever we're delivering is additive and non-competitive binding because these two different binding sites. Our planned trial is to combine these two, so we're able to give a dose that is below a toxic dose with either compound, but deliver much more radiation, in this case, to tumors. And we know there's a dose response, and we also know that more or less the only site of PSMA expression that is targeted by both of these delivery methods is tumor. So we should be able to deliver a lot more with less toxicity. So this is a planned trial for 2018. But as I was talking about the antibody, where we're going forward with that as of a couple months ago, and enrolling the second cohort currently, is using alpha emitters or targeted alpha emitters. So this was the top is a initial 2-patient publication, and the bottom is a subsequent 14-patient publication out of Germany. But we all know, just in terms of properties, that alphas are more powerful, although a smaller distance. Clinically, they've been validated with the approval and current use of radium-223. Those are not tumor target, or that is not a tumor-targeted drug. That goes to stroma that is next to tumor cells and may interfere with the stroma, leading to better patient outcomes. This would be tumor-targeted, and we can see here this is a quite impressive case report. And you can say what you want to say about PSMA imaging, maybe just get rid of the target, but in this case you can see PSA really went down and the patient did well. You can also see that the salivary glands were really ablated, and we know on an anecdotal basis this can be a clinical problem. You can see their PSA waterfall plot, but as in most of the retrospective studies, there's a bunch of data that is missing that's inherent to retrospective studies. So we started 2 months ago, a PSMA-targeted alpha emitter study, but we're using the antibody instead. And the main reason we're using the antibody instead is to basically eliminate the uptake in the salivary glands especially, but also the kidney in case that's a longer-term issue. Is unclear if there will be marrow toxicity. Some people would say that there will be none because of the short range. Some people say there's going to be a lot. So we're starting off with discussions with the FDA as a slow dose escalation study. Like I said, we're enrolling the second patient now, and it's, first cohort didn't have dose-limiting toxicity, and we have studies ongoing, so we will see. But it's nice that we have both alpha and beta emitters in the clinic, and we have both small molecules as well as antibodies in the clinic. And I think ultimately that combination is going to be what's going to move the field forward. As I talked about, the other major aim besides delivering or optimizing the dose that we're going to be able to administer to humans, what we're also looking for is a optimal patient population. We're looking for additional funding for this, but what we have is a big retrospective data set, and we have some prospective data sets for both looking at the clinical parameters as well as some immune parameters, as well as some genomic parameters, tumor genomic, as well as germline parameters that we hypothesize that we'll be able to optimize the patient population for this type of therapy in the future. So basically, that's a quick rundown of kind of the history as well as our current trials. There are prospective studies that are also going on other places in the world. So there's a randomized trial that is going on in Australia right now for late-stage disease, randomizing between lutetium PSMA-617 and the chemotherapy cabazitaxel. There is a study at Memorial Sloan Kettering using MIP 1095 as a small molecule with iodine-131. It's actually the only other dose escalation study of which I'm aware. And there's a study through a company called Radiomedics. It's a two-institution study that's been enrolling for several months in Houston and at UCLA with what I would consider a moderate dose, but they are able to repeat dosing. So besides New York, there's two other cities in the United States where patients will have access to this type of therapy. Editorializing, in New York, we are using funds for that. And in other places, it's from patient funds. But my overall kind of assessment is that I think we've been moving the field forward in a lot of ways, and I would say that finally PSMA-targeted radiotherapy is moving forward in prospective trials.
Scott, that was a great presentation, and thank you. This is an exciting new area, and you're certainly one of the pioneers and leaders here. Kind of reminds me, Star Wars is being released this week, and this is really sort of the Star Wars of the tumor-targeted therapy. At some of those case presentations were really clinically unbelievable. And I'm wondering, just as a question, are you guys working on ways to block uptake in the salivary gland and kidneys that sort of limit the toxicity of some of these agents?
So it's been worked on. So the short answer is yes. What has been done so far is mostly been because most of the patients have been treated so far in Europe, and they're not officially treatment studies. So anecdotally, it doesn't look like flooding the system with peptides does too much. Which is why we have not added that in terms of our trials. It does not look like ice works for the salivary glands. There may be some anecdotal data for something like Botox, as is used in kids with hypersalivation. That's really anecdotal, so we don't really know. So, as I mentioned, our approach is to avoid the salivary glands using antibody. And luckily, our experience and what is the experience as far as we can tell with Germany, but there's also the same thing with prospective experience in Australia, is that with the beta emitter, it's damaging but not lethal, essentially, to the salivary gland cells. So it's low grade and reversible as a general rule. It's much more of a worry with the alpha emitters, and it's probably true in the kidney too. You know, we haven't seen any significant clinical elevations of creatinine or renal failure. The mouse model, however, says that maybe if it happens, it's, you know, they saw it at more than a year. So, you know, it's hard. By that time, they're probably off our study and getting something else, and we know that patients with prostate cancer creatinine goes up sometimes. So it's difficult to say, but part of our deal with the FDA for our alpha study is we're following for 2.5 years after they go off study.
So those of us that have been around remember the beta emitters, strontium and samarium, and the toxicity with those, and that sort of radium-223 was a game changer with the alpha emitter and less penetration and toxicity. So do you think the direction here is with sort of a hook up with a alpha particle rather than beta in the future?
I think, yeah, I personally think, and I know from talking to others that most people think if we had to choose between each one, we're more excited about alphas. I think there will be a place for beta emitters, you know, and depending on what ligand is used, you know, at least they may be more safe. There's been, you know, alphas were preclinically studied, you know, a long time ago, and were kind of abandoned because of their short range. It was said that they wouldn't work for larger tumors, but, you know, I think if we can deliver, you know, with a good ligand or antibody to all the cells, or at least most of the cells that may not matter, and, you know, clearly radium led to an improvement in overall survival without even directly hitting at least some, if not the majority of the tumor cells. So I think their potency is quite exciting, and, you know, my thoughts are more or less if you can get an alpha particle into the cell next to the nucleus, it doesn't matter what genomic changes there. There's essentially no resistance mechanism. The key is kind of delivering it there. So I think that there will be, you know, I'm more excited about alphas, but I think there may be a place for both, and maybe it's going to be a combination of the beta and alpha. You know, we'll see.
Yeah, I mean, and for sure that alpha delivers more punch, double-stranded DNA breaks, and other things like that that occur. This is sort of a sideline. Do you think some of the effect of radium-223 and the Alsimca trial and other trials may have been related to some effect on stem cells at all?
Well, I think that it's relatively clear, as clear as it can be with the available data, is it's not, all the effect is not from direct tumor targeting. So whether it's on some sort of the stroma, whether it's on stem cells that may be nearby, whether it's an immune effect, it may be little bits of all of those different aspects, but there are likely some stem cells that are in those areas.
Scott, this is an exciting therapy, and it generates a lot of enthusiasm with patients when they see complete responses in pretty refractory disease. How do you suggest we go forward with the studies and implementation so we have good clinical trials and outcomes, and hopefully an approval in the U.S. sometime soon?
Well, I think you said the key word or phrase, which are clinical trials. I think we need to really perform this therapy to really assess what is the right patient population, what is the true efficacy, and what is the true toxicity. And the only way to really tell that is in clinical trials. It is nice to know that there are some prospective clinical trials that are going. It is my opinion that this should more or less only be done in a clinical trial, because we can help that individual patient, hopefully sitting right in front of me, as well as society. So that's my opinion. Is that this should always be done as part of that. You know, to enable that to happen, I think that there needs to be additional funding available, and it would be nice to have additional pharmaceutical companies in the mix, it's nice to know that at least one has gotten in a mix with PSMA. Actually, that would be a second one because of the antibody. But we saw that Novartis just bought a smaller company with the same concept for neuroendocrine tumors. So I think that Big Pharma may be moving towards this, and I think that will help support clinical trials to be done at a wider scale and get this done faster.
I know a lot of the companies are trying to get their imaging approved in the U.S. We have Aximum, which is available now, but not everywhere. And PSMA, as you mentioned, there's a lot of excitement about that with we call it the next generation imaging. We just did our Radar 3, which focused on all these new imaging and finding the disease earlier. So again, thanks for your contributions. This was an outstanding presentation.